A very good afternoon, ladies and gentlemen, esteemed guests, and dedicated advocates for climate action. Climate Governance Malaysia welcomes you to the Seeds of Tomorrow, addressing climate challenges with the Svalbard Global Seed Vault. It's an honor to stand before you as your MC for today's event. With climate change threatening the future of life on Earth, this webinar brings together experts to explore the efforts being taken to preserve our precious plant species and their critical role in tackling climate change. We'll begin with opening remarks by architect Dr. Serina Hijjas, council member of CGM, followed by a keynote address from Mr. Åsmund Asdal, Svalbard Global Seed Vault Coordinator at the Nordic Genetic Resource Center, or NordGen. Next, we'll transition to a panel discussion moderated by Dato Henry Barlow, Chair of the Board of Directors of CGM, featuring esteemed local and global experts as panelists. The webinar will then conclude with closing remarks by Dr. Gary Theseira, Chair of the Council of CGM. Without further ado, please join me in welcoming architect Dr. Serina Hijjas to deliver her welcoming remarks. Dr. Serina, over to you.
Thank you very much. Good afternoon, ladies and gentlemen. Climate Governance Malaysia is extremely honored to have you here with us, and very good morning to our esteemed keynote speakers keynote speaker Åsmund Asdal from NordGen, joining us from Norway, which is at 7 a.m. his local time. We also have Dr. Paul, who's joining from London, which is at 6 a.m. his local time. And with us also today, a round of expert panelists, which has been introduced by our former speaker. If society started bending the curve globally when the greenhouse gas emissions were rising and the Kyoto Protocol was enforced, we may have been able to turn back the clock of reaching the 1.5 degree threshold. Instead, today we have surpassed 6 of the 9 planetary boundaries and haven't been able to mitigate the rising emission curve affecting climate change and our ecosystem services. Land degradation, urban expansion, deforestation have resulted in the loss of fertile agricultural land and plant diversity, thus increasing the risk of extinction for many plants and crop species. Loss of wild crop relatives threatens food production because they contain genes for traits such as pest resistance and drought tolerance, which plant breeders can use to improve the performance of cultivated varieties. I have a very small experience through the work of Angela Hijjas, cultivating Malaysia tropical trees in a very small arboretum with over 100 species of Dipterocarps family. 5% of the tree species are critically endangered, 6% are endangered, 10% are vulnerable in the wild, according to the IUCN listings. Seed and gene banks provide extremely important biorepository that serves to preserve genetic material of species that are endangered or close to extinction. Shortly after the enforced 2005 Kyoto Protocol, Norway had the foresight to establish the Svalbard Global Seed Vault in 2008, a growing repository which our keynote speaker, hero of seeds, Åsmund Asdal, for the Nordic Genetic Resource Center, NordGen, will be speaking about. Thank you very much. Over to you.
Thank you very much, Dr. Serina. Now, let's continue with a keynote address by Mr. Åsmund Asdal, Svalbard Global Seed Vault Coordinator for the Nordic Genetic Resource Center, NordGen. The floor is yours, Mr. Åsmund.
Thank you very much. Thanks for inviting me to this webinar. I'm honored to be able to speak to an audience in Malaysia. And thanks for these introduction words, and also thanks for a very nice introduction video that you had made as an introduction to my speak. So my name is Åsmund Asdal, as already said. I'm employed at Seed Vault Coordinator at the Nordic Genetic Resource Center which is the gene bank for the 5 Nordic countries. Next, please. My first slide just states the fact that the Svalbard Global Seed Vault is the largest backup of seeds of importance to the world's food supply, and I will explain more in detail what this means. Next, please. For this audience, I think it's not needed to explain more in detail why we are conserving crop diversity, but just for the records, I just repeat that plant genetic resources is the raw material for plant breeding and development of agriculture and food production. We need this genetic diversity, not at least to adapt farming, to adapt food production to climate change, which is one main issue for this webinar. Next, please. There are two aspects related to the global seed vault when it comes to climate change. The first point I will make is that the plant genetic resources that are conserved in the seed vault are crucial for adapting to climate change. Plant breeders, scientists need these plant genetic resources, and in a changing climate, these resources are more valuable and precious than ever. On the other hand, I'm also often asked what will happen to the seeds in the seed vault when we see climate change and warmer temperatures in the Arctic. The answer to this is that the seed vault construction is artificially cooled and not vulnerable to warmer climate in the Arctic. And as explained in the video, if the cooling system fails, there will still be permafrost down to -3, and it was said that the temperature would need weeks, perhaps months. Actually, it took 1.5 years to cool down the seed vault from the permafrost level at -3 down to -18. So it will probably take around a year before the temperature is on the same level as the permafrost around, because a huge mass of the soils and rocks around the seed vault is also cooled down to a very low level. So we feel that it's quite safe now, and we will have months to replace a failed cooling system if something happens to that one. So we feel that climate change is affecting the Arctic quite severe, yes, but the seed vault is pretty safe also to keep the seeds frozen even if the temperature gets warmer in the Arctic. Next, please. The first seeds in Svalbard was placed in a coal mine in November 1984. This was a backup of the seed samples in the Nordic seed collections. This was a brilliant idea. The management of this Nordic Gene Bank found this coal mine with permafrost and brought backup samples of the Nordic collections there. And this solution gained a lot of attention worldwide, and that was how the idea of storing seeds in the Arctic initially was launched. Next, please. A brief history about the global seed vault. Many gene banks have lost seeds, and the need for a global safety facility for seed conservation has been discussed already from the 80s and 90s, actually. When we had the international treaty in place in 2004, it was discussed to reassume discussions about having a seed vault in Svalbard. The seed vault was the decision to build the seed vault was announced by the Nordic prime ministers in 2006. The seed vault was built in 2007, and it opened, and the first seed were taken in February 2008. It's actually 17 years ago now. Next, please. So the main objective of the seed vault is to conserve security samples of seeds conserved in gene banks. We also quite soon discovered that the seed vault became quite symbolic, quite iconic symbol of the importance of taking care of genetic resources. So we use the seed vault quite deliberately to increase the public awareness about this objective. And we also feel that the seed vault, one of the conditions for storing seeds in the seed vault is that the genetic material should be available for plant breeding and research, and by that we also contribute to keep genetic resources in the public domain and not be privatized, patented and privatized. Next, please. Gene banks are invited to send copies of their seed samples in the seed vault. It's free of charge to put the seeds there. The gene banks are the owners of the seeds that are stored there, and the seeds can be returned only to the owner. We often get requests for sending seeds that are stored in the seed vault, but we always tell them to approach the gene bank that is the owner, because this gene bank have the same genetic material, and by signing the deposit agreement, this gene bank has committed themselves to share the genetic material to breeding and research. Next, please. The seed vault is built and owned by Norway. It's managed and operated in a partnership between the Norwegian Ministry of Agriculture and Food, the international organization Crop Trust, and my organization, which is NordGen, the Nordic Genetic Resource Center. Seed deposits are handled by us in NordGen, and we do not stay in Svalbard permanently, but there are staff in the governmental bureau called Statsbygg. They look after the facility. They do not do anything with the seeds, but they just look after the facility, that temperatures are fine, that equipment works, and that no one breaks in and so on. Alarms and surveillance systems are managed by Statsbygg. Next, please. And then we have this international advisory panel, and they will have a meeting in the end of this month. They make an inspection in the seed vault, and they discuss topics that are relevant for overseeing and giving advice for the management of the seed vault. And luckily, we have one of the attendants here, Dr. Bin Ghazalli. He will go to Svalbard in the end of this month and take part in this important meeting. Next, please. Just one slide about the Nordic Gene Bank. It was established in 1979. It turned into the Nordic Genetic Resource Center in 2008. And the objective is to conserve and distribute genetic resources for the Nordic countries. Nordic material that is relevant for Nordic science and Nordic plant breeding. For the time being, we have around 35,000 samples of more than 500 different plant species and subspecies. Next, please. Here you can see a model of the seed vault. It's built in solid rock. It's not in a coal mine. That's important. It's a common misunderstanding. And as already said in the video, the permafrost here provides -3, -4, and it's artificially cooled down to -18, which is the recommended level for long-term conservation of seeds in gene banks. And of course, there are quite advanced monitoring and surveillance systems there. Next, please. We have 3 seed chambers. One of them is filled up with, each of them have the capacity to store 1.5 million samples. We are these days, and now in the end of February, we will bring in seeds from around 20 different gene banks. Among these, your MARDI gene bank. We'll bring them in, and we now use chamber 1. Chamber 3 is still not used, so we have plenty of space to store more seeds. Next, please. We organize what we call seed vault openings 3 times each year. At these occasions, we invite gene banks to send seeds, and then we go to Svalbard and we bring the seeds into the seed vault. Gene banks, they provide accession lists with some basic data about all samples that are conserved in the seed should be conserved, and these data is uploaded to the seed portal database. Next, please. This is the interface of the seed portal. Anyone can enter this and see how many seed samples that are in the seed vault at any time. As of today, there are 1.3 million seed samples. More than 6,000 species are represented, and 103 depositor gene banks have already sent seeds to the seed vault. Next, please. Here you can see the number of seeds that have been deposited in the seed vault over the years. Started with around 300,000 samples in 2008, and the large gene banks, international centers, and large national gene banks, they sent major parts of their seeds during the first years, and then we have had a quite steady increase of seed samples inside the seed vault to be deposited each year. You can see some drops of the curve here, and that's I will come back to that, but that's the years when the International Center of Agricultural Research in Dry Areas, ICARDA, they retrieved the seeds they had deposited from Aleppo in Syria. Come back to that. Next, please. From the seed portal, the website, you could do this yourself on your computer, but I extracted some data that are relevant for Malaysia. Today, 5,897 seed samples are of the origin from Malaysia. 111 species from are represented in this collection from Malaysia, and all these seeds are deposited by 16 different gene banks. Most gene banks in the world, they have samples from many different countries. Some of the centers have seeds from more than 100 countries. That's why there are 16 different gene banks that have deposited seeds from Malaysia. And the first MARDI deposit took place in May last year. 207 samples of rice were deposited at that time, and we will have new seed deposit now in February 2024. 508 samples of rice, solanum, and Vigna beans will be deposited there, and Dr. Ghazalli will be invited to deliver these seeds at the seed vault entrance during a seed deposit ceremony. And hopefully we will have some nice photos, and you will see them on your TV and maybe newspapers in Malaysia, so the public can see that your country is also taking advantage of this opportunity to store seeds in the seed vault. We look forward to that. Next, please. And I'm just having a couple of slides related to conservation of material forest genetic resources in the seed vault. The seed vault can only conserve orthodox seeds. Non-orthodox seeds cannot stay for a long time in conditions like this. So plants that do not produce orthodox seeds, they should be conserved by other methods and not in the seed vault. For the time being, the seed vault is dedicated to plant genetic resources, and one condition is that the material should be available for breeding and research according to regulations in the International Plant Treaty. So one of the differences between plant genetic resources and forest genetic resources is that we have this international treaty for plants, crop plants, but we do not have a similar system for tree plants, pure tree plants. The plant treaty provides a set of rules for conservation and use and exchange of plant genetic resources. We have a standard material transfer agreement that is used when gene banks distribute seeds to users, and we also have a multilateral system for sharing information. We do not have the same system in place for forest genetic resources, which is one of the reasons why the seed vault is not generally open for conserving of tree seeds, forest genetic resources. Next, please. However, there is an unclear distinction between plant genetic resources and forest genetic resources, especially in tropical trees and tropical and less industrialized agriculture. In many countries, trees are considered to be plant genetic resources because they have functions in intercropping between food crops, soil stabilization. They are used for animal feed, for honey production, charcoal and energy for cooking and cultivation, pulp and fiber, and timber used for agricultural tools and fences and buildings. So these are reasons why many gene banks consider trees to be also plant genetic resources. And there are, for the time being, many seed samples of trees in the seed vault. So this restriction, or what we should call it, is not very strict. So if a gene bank consider a seed sample and a species to be a plant genetic resource species, we of course accept that. And having said that, the International Advisory Panel, where Dr. Ghazalli will attend, will also discuss if the seed vault in more general should be opened to conserve seeds of forest genetic resources species. Next, please. We have an agroforestry World Agroforestry Center in I think it's Kenya. They are a regular depositor of the seed vault, and in total they have 1,980 accessions of 188 different species belonging to 105 different genera of different tree seeds in the seed vault already. So just to explain to you that we are quite open to also accept tree seed species in the seed vault. Next, please. Yeah, here is a list of seed samples from World Agroforestry Center. You see the genera here, and you also see the number of accessions. For instance, acacia here, they have 396 different accessions belonging to 26 different species of the genus Acacia. So, and as I said, there are 105 different genera in the represented in this seed collection from the World Agroforestry Center, and so yeah. The conclusion is that if a gene bank wants to send tree seeds and consider this to have some relevance to the plant genetic resources sector, we accept to include them in the seed vault. Next, please. Then we have one example when a gene bank withdrew seeds from the seed vault. I noticed during the video that two names were used. It was called Doomsday Vault. Actually, we do what we can to avoid using this. Media and journalists love this expression, this term, but it's not a seed vault that is meant to be used. in a remote future after doomsday. It's more an active part of an active international system, cooperation for conserving and distributing genetic resources of plants. The video also used the Noah's Ark, and that's a much better expression. It explains that seeds are stored, taken care of, and they are available for use here and now, and not just after doomsday. So Ikada, formerly located in Aleppo in Syria, luckily they had deposited 116,000 seed samples in the seed vault between the opening in 2008 and 2014, which is almost the whole collection they had in Aleppo. Due to the civil war in Syria, they decided to move the gene bank activities to Ikada units in Lebanon and Morocco, and we sent all the deposited Aleppo seeds back to these countries, these units, in 3 portions in 2015, 17, and 19. And all the seeds arrived, all the seed samples germinated, and this is an excellent example on how the seed vault could work. If a gene bank needs the seeds to be returned, we will send them back immediately, and the gene bank will be able to restore the gene bank activities based on the seeds that they deposited in the seed vault. So this is tragic for Ikada and for Syria, of course, but for the seed vault, it really displayed and showed the importance of having an international facility like this. And again, more than 100,000 samples have been produced again by Ikada in Lebanon and Morocco, and they have deposited more than 100,000 samples back to the seed vault. So this is really a success story showing hard work by Ikada, good decisions for backing up their seed collections, and also hard work to re-establish a gene bank that was destroyed. Next, please. Yes, then just a few photos showing the process and what we are doing. Seeds are normally packed in watertight aluminum pouches like this, already explained through the video. Next, please. Seed pouches. Yeah, here we see all these seed boxes. Seed samples are packed inside boxes, sealed boxes. We do not open the boxes in Svalbard, but we scan all boxes in the security system at the airport to be sure that there is nothing else but seeds inside the seed boxes. So here you can see how boxes looked like when we do not open them, but we scan them to see what's inside. Next, please. Then after scanning, security scanning at the airport, we bring all the seed boxes to the seed vault. Next, please. And the weather there can be really harsh. This is from February 23, when we had to carry the boxes to the seed vault. The trucks couldn't go because of snow. Next, please. We put such labels on with some basic information and also relation connection to the seed portal database. So at any time we can see which are the boxes, where are they placed in the seed vault, and what is the content. Next, please. Then we bring the seeds to the shelves. Next, I see that my time is out now, but I'm ready in a second. Here you can see a diversity of box shapes and colors, materials, which is very inspiring to see all these different boxes in the seed vault. Next, please. Yeah, that was my last slide. Thanks for the attention. This is the smile you can have when you, after a week of work, can state that the seeds have been secured in the seed vault, and everything went well. During all these years, not a single box, not a single seed sample have been lost on their way from a gene bank to the seed vault. Thanks a lot.
Thank you for the engaging keynote, Mr. Åsmund. Ladies and gentlemen, before we move on to our next session, we kindly request you take a few moments to complete our survey. Your feedback will help us improve and deliver even better experiences in the future. Thank you. We now proceed to our panel discussion, which will be moderated by Dato Henry Barlow, Chair of the Board of Directors of CGM. He will guide the discussion alongside our 4 esteemed panelists. Dr. Dzaeman bin Dzulkifli David, Executive Director of the Tropical Rainforest Conservation and Research Center, TRCRC, Dr. Muhammad Norfaizal bin Ghazalli, Deputy Director for Malaysian Agricultural Research and Development Institute, MARDI, Dr. Paul Smith, Secretary General of Botanic Gardens Conservation International, BGCI, and Dr. Zainal Zahari Zainuddin, Program Director for Bringing Back Our Rare Animals, BORA. If you have any questions for our panelists, please use the Q&A function at the bottom of your screen to send in your questions. Dato Henry will address as many of them as time permits during the discussion. Please join me in warmly welcoming our esteemed panelists as they lead us through this discussion. Dato Henry, the floor is yours.
Thank you very much indeed, and welcome to everybody who is taking part in this webinar. I hope you find it instructive. In my initial exchange of emails with Åsmund Asdal, and in his talk today, stress was laid on seeds of importance for feeding the world, which are stored at Svalbard seed vault and indeed elsewhere. In this forthcoming discussion, I'd like to broaden the issue from a Southeast Asian perspective. How to ensure that in the face of climate change, we can retain the enormous plant biodiversity of the Southeast Asian rainforests, bearing in mind that a high proportion of Southeast Asian rainforest plants have recalcitrant, or also known as non-orthodox seeds. These are seeds which cannot be frozen to retain their viability long term. And let us also not forget that the leaf litter and topsoil in primary rainforest forms the habitat for innumerable insects, fungi, and other microorganisms, many of which have not been described scientifically. So without further ado, I would like to start the discussion by requesting Dr. Faisal of MARDI to give a presentation. We hope that each discussant will be able to produce a presentation of about 10 minutes, which should leave us time at the end for a little discussion.
Okay, thank you, Dato. Okay, I'm Faisal from Agrobiodiversity and Environment Research Center. Okay, and in this center, we have cater for some types of the conservation and also bioprospection of the genetic resources. So when we talk about the conservation, we need to require something that need to be bring back to the ex situ conservation, because sometimes we need to cater for the farmers and also from the stakeholders and also the ministry needs. Okay, right? In the gene bank, we have 2 facilities to cater for both orthodox and also recalcitrant seeds. Okay, we have the certain station in Terengganu, in Negeri Sembilan, and also in Serdang is the National Center Collection, cater for the more genera, consists of rice, rare fruits, vegetables, tubers, and also palm fruits. Okay, some of the seeds can be conserved via ex situ in the seed bank, and of course for the recalcitrant seeds, we need to conserve it via field gene bank. Okay, and for the gene bank, we have some limitation because of the manage to the fund and also regarding the labor works that need to be catered from time to time, right? But we are grateful by the support by the government and also by the certain agencies from the worldwide and also from the local agencies, from the Ministry of the Natural Resources and also from the Ministry of Agriculture and Food Security. We managed to get the fund to manage these genetic resources. Okay, to date, we have conserved around 90,000 accessions from the rice, traditional vegetables, fruits, and also agrobiodiversity related to the food security issues cater from the 1980s collection. Okay. For the BOL project, stand for biodiversity for opportunities, livelihood, and also development, funded by the government of Norway and led by Crop Trust. We managed to jump into the boat to conserve the seed into Svalbard. Okay. To date, we have deposited around 725 accessions of rice, long bean, and also eggplants, which is important to the culture, to the unique characteristic. Okay, some of the characters for the rice, I can say that we collected from the villages and also from the orang asli or orang asal communities, which cater for the certain unique characteristic like the scented rice, colored rice, and also the green shapes. Okay, we need to preserve this genetic resources to make sure that we can use it in the future and to get the best thing that we managed to make it in the first tier and also second tier the application in the respected gene banks such as ERI, Wolvish, and also in the My Gene Bank. Okay. In MARDI, we have two types of gene bank. For the seed bank, we have the main facilities in Sprang Prai, Penang, and to date we have around 13,000 specimens. Most of them consist of rice and also traditional vegetables, and also we also conserve the crop wild relatives in partnership with the Millennium Seed Bank in 2016 to 2018 base project. Okay, when we talk about the conservation, there are some pertinent issues that we in MARDI and also those who are related to the conservation need to think. First is to find the gap fillings. Okay, how is we need to select the genus or the genera that need to be conserved in the gene banks, specific or the targeted collections, and also we need also to cater the recent needs or the issues of the loss of genetic diversity from the genetic erosions. Okay, and the basic elements that we in MARDI cater is the land races, the ancestor of the crop wild relatives, and also the weedy type of the cultivated plants. And these are the mandate that we need to cultivate from time to time to make sure that we can get as much as possible the precious genetic resources conserved in our collections. Okay. So we also facing some challenges in managing this collection, like the pests and diseases, and also the problem of germination, and also maintenance of the replanting, and also in maintenance the laborious works from time to time. I think that Dr. Dzaeman will elaborate later regarding this because he's more on the TRCRC. Okay, and regarding the forest genetic resources, FGR that presented by Åsmund, okay, I think that it is important because for now we're trying to jump into the cryopreservation study on the recalcitrant streets involving certain genera like Garcinia, Bacauria, the wild mangosteen, and also the wild mangifera, wild mangoes. So, okay, that is the thing that I think that MARDI can share from time to time and train and also to make sure that all this agro-biodiversity is conserved precisely and also sensitively in the terms. Okay, I think that Dato, maybe that is the introduction from myself. Later we can discuss later based on the question by the audience and also by the panels.
Thank you very much indeed, Dr. Faisal. May I now call upon Dr. Paul Smith of BGCI to deliver his introduction.
Thank you, Dato Henry. Okay, so I'm going to talk a little bit about forest genetic resources and about trees, particularly those that occur in Malaysia. Now, my organization, Botanic Gardens Conservation International, is a consortium of about 900 botanic gardens and arboreta around the world, and we lead the global tree assessment for the last 10 years, which is now more or less complete. And that means assessing the conservation status of all 58,000 of the world's tree species. This has been a huge undertaking with over 1,000 contributors from around the world. And one of the results of this is that we are able to, we publish checklists for every country in the world. If you go to BGCI's website or you Google Global Tree Portal, then you can select a country and you'll be able to download checklists of indigenous tree species for those countries. So Malaysia has 5,282 tree species in total, and of those, 1,378 are endemic, so they only occur in Malaysia. And of those 5,000 odd tree species, 1,211 are threatened with extinction. So that's around between 20 and 25% of Malaysia's trees are threatened. Now, critically, 74% of these of Malaysia's tree species are not in ex situ collections. So they're not in seed banks, and they're not in field gene banks, not in arboreta or botanic gardens. The figure for in situ is a little better, but still 51% of Malaysia's tree species are not protected in situ in protected areas. So that's a big chunk of tree diversity which has no formal protection. We have a lot of work to do. Dipterocarpaceae, of course, the dipterocarps are keystone species in Malaysian forests, and we're fortunate that we also have a very recent study of the world's dipterocarps that was published in December 2022. There are 535 species of dipterocarp globally, but Malaysia and Indonesia are home to the largest diversity of dipterocarps. And 67% of all dipterocarps are threatened with extinction. And of these, 70 are assessed as critically endangered, 143 are endangered, and 144 are vulnerable. There's one documented extinct species, that's Hopia shinkang. A further 22 species are data deficient, so we really don't know enough about them. And they may even not still be with us. They may not be extant. So amongst those dipterocarps, 201 are single country endemics, of which 84% are threatened in the wild. So again, a very high proportion are threatened. If you take dipterocarps as a whole, 71% are protected in situ, so in protected areas. And that means in combination with management of these sites and production forest, monitoring, habitat restoration, and rehabilitation are also recommended conservation actions, even though they have a level of protection. But if you take Dipterocarpaceae as a whole, 64% are not in ex situ collections. So we don't have a backup like Svalbard or any other seed bank or gene bank collections. So we have 194 that we know of that are in ex situ collections, and these are established in botanic gardens, parks, nurseries, seed banks, germplasm collections, or arboreta across the globe. But most of those species are only recorded in one collection. So that means they're not genetically diverse. So 103 of those 194 are only in one collection. 2 species are found in more than 10 ex situ collections, but for the rest, there are very few actually duplicates in those collections. So the big question is, what can we do about this? And this is really, I think, where we want to focus our discussion. And we'll talk about restoration later on in this session. But there are a number of things that we can do. But the first thing is to protect as many of these tree species as possible in situ. And the great thing about having the global tree assessment data is it's geo-referenced. So we know where these trees are. We have records of where they are. So we're already seeing Sabah Forestry Department using the new global tree assessment data to designate areas of high conservation value, where oil palm plantations and so on are no-go areas. And the latest assessment, which I think was carried out last year, was based on more than 300 threatened species of tree. The previous version was based on just 28 threatened tree species. So having more information about threatened species is hugely helpful in ensuring that we protect what we have in situ, where it really needs the most protection. The second thing we can do is conventional seed banking for orthodox threatened tree species. And as has already been explained, orthodoxy means desiccation tolerant, where you can dry the seeds down, you can store them at -18, -20, and many of them live for decades and if not centuries. Around 70 to 80% of the world's plant species produce orthodox seeds. The rest are what we call recalcitrant or intermediate. You cannot dry those without killing the seed. Now that ratio goes up hugely in tropical forests. So dipterocarps, for example, are recalcitrant. You can't dry them and store them in a conventional seed bank. So the answer there is field gene banks. And field gene banks have been used in agroforestry for decades. And field gene banks are simply living collections of plants that are kept and maintained. Those are used for fruit trees and so on. But in the case of conservation, where foresters will choose plus trees that have characteristics like a straight trunk or nice fruit or whatever that they want to preserve, in conservation, we need to make sure we are conserving the genetic diversity, the wild genetic diversity of those trees. So field gene banks for conservation purposes will be artificially planted gene banks, but with highly genetically diverse trees within them. Now, one of the things that BGCI has done over the last 5 years is to set up global conservation consortia for these recalcitrant and what we call exceptional species. One of those is Dipterocarpaceae. So we have a global conservation consortium for dipterocarps that involves 25 different partner organizations across Southeast Asia. And what they do collectively is to make sure that we have all threatened dipterocarp species in ex situ collections and preferably in field gene banks so that they're genetically diverse. Now that process is still at the beginning, but that is the ultimate aim, is to get every dipterocarp species into field gene banks so that they don't become extinct, so that we don't lose their genetic diversity. And this is about meta-collection management. So if you like, the collections are managed collectively across the countries, across geographic gradients. And that's useful for understanding or climate resilience, understanding the horticology of species. So which soils do they prefer? How susceptible are they to different climates and so on. And that helps us with understanding how we can also reintroduce and restore those species. It also gives us propagation protocols. So how do we actually cultivate those species? And then most important of all, a source of diverse material for reintroduction and restoration. And that's where I will leave it for now. Hopefully Dzaeman can pick up the story around how we use that material for restoration. Thank you.
Thank you very much indeed, Dr. Smith. And now we go on to, I've lost my paper. Here we are. Third speaker is Dr. Dzaeman of TRCRC. Dr. Dzaeman, over to you.
Hello, everyone. Thanks for having me to Climate Governance Malaysia, and great to share this floor with all these esteemed guests. My work actually at TRCRC, or Tropical Rainforest Conservation Research Center, actually goes back to when we first started, and actually when we first met Paul when he was head of the Millennium Seed Bank. And there, what we discussed was basically how do we preserve this. The board members back then were very interested in setting up a Malaysian seed bank, but quickly were demotivated by the risk and by the cost that is required to do so in this region. So at TRCRC, what we're focusing on right now is ex situ collections as one of our core programs. So living collections or field gene banks is what we do. And we're setting them up across the different regions, because within Malaysia, we have very different sort of microclimates, we have different sort of terrains, we have different sort of soil types. So the key is basically how do we then now create these living collections, or field gene banks across our nation, and taking into consideration the different conditions that we have. So in Sabah, we have one, in Perak, we have one, and right now in the urban setting in Selangor, we have one. And these different sites are quite large scale for gene banks per se. They're about anywhere from 200 hectares to about 500 hectares. As we develop these sites, what we're doing is not only conserving them, but actually actively using them in restoration as well. So instead of seeds for tomorrow, actually the topic will be more seeds for today. What is very difficult in diplocrops, not only that their seeds cannot be stored, is that they also cannot be attained, or you cannot get them as frequent as you need them. So the diplocrops fruit and flower every 5 to 7 years in synchrony with El Nino. And what happens is the seeds fall and germinate almost instantly. Within 2 to 14 days as the germination rate. And so the teams required to actually do this work in those 3 different sites will need to now first map out where all the seeds are, follow fruiting and phenology, basically fruiting and flowering in the state, and then how then you prioritize different species, because different species will have different level of threat to them, and so we need to prioritize them, especially when they're all falling at the same time. And one example was during the COVID period, when actually we couldn't go out and collect. We actually mobilized the different communities living next to forests in the urban area. We tested this out, and we found that actually we were able to collect seeds from such restricted amount, and actually filled up our nursery till it was full during the pandemic. And this was an amazing feat to show that we can get together and really scale up this. So then from the last 10 years, we were developing the protocols, we're developing methodology and the sites that we work on. We're now at the stage of how do we then scale up? And scaling up requires a lot of technical ability, and also requires a lot of manpower. So here we are now developing an application that can actually carry out all the work that is needed to with your handphone. So you won't need a GPS, you won't need a clinometer, for example, to measure the height of a tree. You won't need a lot of these tools to measure where your seeds are coming from. So with these protocols and with these apps, we are hoping to then scale up to the rest of the communities that are using restoration or carrying out restoration. What is important is to scale up and to make sure that we're restoring with the correct species, at the correct space, because with restoration, there's many different positive side effects. So you can improve with biodiversity, we can conserve different species while restoring. We can also then at the same time protect watershed, and we can also then work on carbon sequestration as well. And so there's a range of different positive things that you can do with seeds right now. And this is where we're heading to. How do we scale up and how do we do it correctly by minimal cost and not bottlenecking the whole system? And I think hopefully during the discussion, we can talk about some of the tools that BGCI as well has created. So we have the Global Biodiversity Standard, which helps ensure that when you carry out restoration, you're carrying it out with the correct methodology. Okay, thank you very much.
Thank you very much indeed, Dr. Dzaeman. Can we now go on to our final speaker, Dr. Zainal of BORA. Over to you, Dr. Zainal.
I believe Dr. Zainal, you're muted.
Yeah, Assalamualaikum and good afternoon. In BORA, most of our enrichment is focused on habitat restoration for the orangutans using the genus Ficus. And these are done inside oil palm plantation and forest landscapes. The reason why we use Ficus, or commonly we call figs, or in Malay is called ara, nunuk, and bringin, etc., is because it is a favored or preferred orangutan food plants. And they would consume the leaves and the fruits as well. But again, in addition to that, ficus also forms an important diet to other wildlife species. And we talk about the whole biodiversity. And they rely on ficus as supplementation of their diet when the deep throat carp are not fruiting. So it's important for species like the hornbills, the other non-human primates like the gibbons, and even wild boar, which is the bearded pig in Sabah, the samba deer, the Bornean bunting, and even elephants. They do consume ficus species. And with that, we know that ficus species is an important keystone species. And other reasons that as to why we chose ficus are one, they are fast growing and they fruit many times a year. And when they fruit, the yield is enormous. You can see the whole canopy is filled up with ficus fruits. This is important. The second reason is that there are 150 species of ficus in Sabah. We have in our collection about 95 species now. And with that 150 species comes the whole diversity. So you have ficus ranging from epiphytes, from shrubs, from trees that go beyond 40 meters tall, from stranglers, and also the unique earth figs, where the fruits are on the forest floor. So these unique characteristics of figs are very important. And also it provides us with a selection of what is needed in which area. So we can really select from all those species that we have. And thirdly, the species is orthodox. So that is the thing that's so important. And to date, we have saved many species. I'll talk about it later in ultra cold freezer, -82. And lastly, the propagation of ficus is so easy. It's not rocket science. You can do it by seeds or even by vegetative propagation. At Sabah Ficus Germplasm Center, where we are, it is inside Tabin Wildlife Reserve. And here we have a plot that's about 3 hectares. And within the plot, we have our living specimens of all those 95 individual fig trees. And at the same time, we have got 2 nurseries where everything is done in-house. So we produce our own fig seedlings. So 2 nurseries, and we also have a ficus quarantine station, where when we collect from outside of Tabin, it goes through a simple quarantine before it ends up in the germplasm. With the Malaysia Book of Records, we hold the largest living collection of local figs in Malaysia, and maybe even in the world. We have 5 staff working there, and the work at the germplasm is, everything is in-house. we produce from seeds to seedlings to restoration. So everything is done in-house. Some of the seeds, when in season, we purify the seeds, we dry them, and we freeze them. So these seeds could be used when the parent plants are not fruiting. So we have all this backup so that we can continuously produce seedlings as and when required. So we do also viability tests of those frozen under ultra cold freezer. So currently we have a viability of between 70 to 86%. And again, there are various parameters that we look at. And we have about, currently we have 45 species of figs that is being preserved under minus 82 degrees Celsius. And once the plant once the plant has hardened in our nursery, then it goes off to be transplanted into the set-aside areas within oil palm plantations. And this again is done by our own staff. And maintaining of plant is quite straightforward. Every plant is tagged using a GPS software, and we know exactly where they are in the field. So we could monitor them, and these are monitored every 2 months, and they are maintained every 2 months to ensure that there are better survivability. And of these, 10% are assessed for growth rates, for plant health, as well as mortality. All these databases are kept within BORA. And we do also field research, which again is related to the growth rate, site suitability, which fico species adapt well to which kind of environment. And we try to mitigate some of the field problems because we are still learning on which is the best way to plant whichever species of that 95 species that we have. And at the same time, we also do research in the germplasm, whereby with the limited number of staff that we have, we try to expedite things so that it becomes easier with less workforce. For instance, we reduce the time from seeds to cultivating them in situ from 5 months to 3 months. So that is important for us because we managed to cut short certain processes. The same thing with marcots. We do a lot of marcots as well, or vegetative propagation. Again, we cut short the time from when we harvest the marcots to when we put them in the ground. So these are simple research that we do. And in addition, although we started restoration work back in 2019, but the more extensive work started in 2021 and currently. So now we have major plantations, 9 plantations that are working with us, together with 17 estates. So we have KLK, we have Guthrie, Genting, Sawit Kinabalu, Tradewinds, Glenealy, Kretam, MPOB, and Tiansang. So all those are in partners working with BORA on restoration work. And in total, we have restored about 165 hectares, comprising about 8,500 figs and about 1,000 non-fig species. At the same time, we have provided about 13,000 seedlings to the plantation because they want to plant on their own. So we provide them with those seedlings. And in terms of mortality, it ranges really because we are planting in the open. So it ranges from 8% to 98%. So again, the reasons are very clear-cut. Sometimes it is weather, sometimes it is the monkeys, the macaques, sometimes the weeds. So that's the reason why I emphasize the every 2 months maintenance for whatever we planted. And a lot of the set-aside land that was given to us by the plantations are not the best ones. So we get areas, apart from the riparian, we get areas on steep slopes, we get rocky areas on the hill, We get areas that are prone to seasonal flooding. But most of the time we try to manage and we adapt with the 95 different species that we have. And there are some species that are flood tolerant. So again, we use those in areas where you need species that are flood tolerant. And some species are adapted to rocky, sandy, hill kind of vegetation, and we adapt those plants to those. So far, it is working well, but we are still in the learning process. So it is going to take more time to really get a clear understanding of the species that we plant. And soil, again, in situ, the soil can range from clay, loam, sandy, rocky, or the mixture of all those. So these are also the challenges when we start putting in our plants on the ground. And there are two books that we published in 2024, I think. One is called Orangutan Food Plants. So it again talks about ficus species. And the other one is Habitat Restoration for Fruit-Eating Wildlife. So within this context, I think two things that I've sort of answered. One is the importance of having a gene sort of a field bank or a germplasm where you have your actual living specimens. And the other, with figs, we can freeze them. I think, yes, I will end here and maybe we can discuss much more later. Thank you, Dato Henry.
Thank you very much indeed, Doctor, for your very stimulating account of how you're preserving figs in Sabah and Borneo generally. I think we now have quite a reasonable amount of time to have a general discussion, taking questions which have been sent in from the floor. My hand phone is not very good at picking up these questions, so could I ask our MC to take a judgment call on which questions appear to be the most popular and let us know, and then we'll ask the panelists to answer these various questions, please.
So there's a question by Sean. Are there any samples of seeds taken for reforestation or growing so far? How do we ensure that these seeds are still alive and viable for planting?
Is this addressed to anyone in particular, or?
No, it's not addressed to anyone in particular.
Well, may I suggest that we ask Dr. Dzaeman to answer first, please?
Yeah, so one example of a species that actually we have more in ex situ than in the wild is Vatica cantadensis. It's a limestone species that actually was recently discovered only in 2014. And as it was discovered, it was already critically endangered because of its narrow range. So the limestone company was, or the limestone hill was under threat by limestone mining for cement industry. But we actually managed to safeguard and collect, and now we have over 1,000 individuals outside of its natural habitat. Its natural habitat is close to estimate around 50 or so. We're at the stage now where we are working together with the landowner. They're setting aside the area to as a park. But what we want to do now is that to start to reintroduce them back into the area. So that's just one example. I think we might have more examples from the more different seed banks around the world.
Thank you very much indeed. Any of the panelists got anything further they'd like to add on that? Because if not, may we go on to the next question, please?
So the next question, also from Sean. Any samples of seeds taken for, oh, sorry, that's the same question, sorry. This is a question from Awang Ahmad. Due to the sensitivity and fragile factors, are there any ideas for the best effort or encouragement that can be used to conserve, preserve, or stall the recalcitrant seeds for a long time?
Maybe Dr. Paul Smith is the right person to direct that one to.
Yes, as we explained, the approach for recalcitrant species is field gene banks, and what that means is essentially growing the tree, and then making sure that the tree is protected through to maturity, collecting seed from that, and then using that seed to propagate material for reintroduction. As I said earlier, the best way to approach this from a conservation perspective is to make sure that you are collecting genetically diverse trees. So in other words, you don't go into the field and make all of your seed collections from the same mother tree. You need to be collecting from a large number of individuals, and ideally across geographical and climatic gradients. And also we do temporal sampling as well. So it's a good idea to collect early in the fruiting season, in the middle of the fruiting season, and late in the fruiting season. And that way you get as much genetic diversity as possible to establish your field gene bank. And of course, it's possible to test the genetic diversity of the trees that you have. So unlike in forestry, where you probably want a tree that has certain characteristics, and you probably want to be cloning that tree or using relatively little in the way of genetic diversity for conservation purposes, because we want to reintroduce and because we need to make sure that it's genetically resilient to pests and diseases, to climate, and so on. We need to make sure that these field gene banks are as genetically diverse as possible.
Thank you very much indeed. Any comments from other panel members?
Dato,
yes.
I just want to add on the Dr. Paul. Okay, regarding the species that we conserve, especially for the recalcitrant seeds, some species that have challenges in managing their germination and also their growth, especially those species from the wild durio, and also some species of the mangosteen, wild mangosteen. Okay, and then regarding this, we try to conduct the conservation via cryopreservation, but for the long-term conservation in the lab. But unfortunately, that the process is still ongoing, and we are still try and error in the lab by our team, especially for the Garcinia, because the seed physiological factors is also very challenging. Okay, because Garcinia is some sort of gummy species, and it will have some, I can say that, very challenging in doing the cryopreservation, unlike the Bacauria and also the Mangifera or the Lepidanthe species. But this need a very, I can say that, very costly in doing the research, and we are still preferring to conserve via the germplasm or the field bank. Okay, because but in MARDI, when we conserve it via germplasm, we need to ensure the growth is in a very suitable way. Okay, because from our experience, those trees, when it reach around 20 to 30 years, the age of some species like the wild Mangifera, the productivity of the trees is reducing, and also we also have some problem with the termites and so on. So we need to ensure that those collection is the seed is collected from the same trees, and to back up from the gene bank. And after 20 to 30 years, we need to replanting it back. That is the way that gene bank need to ensure from time to time, and it is also very costly in doing this kind of work without the persistent continuous funding from the ministry. I think that is that can add more on the Dr. Powell experience regarding this forest genetic resources, and also Dr. Dzaeman. I think we can share a lot about this experience later. Okay, thank you.
Thank you very much indeed. Next question, please.
This is a question from Eric Joseph. Is there an opportunity to integrate different species with landscapes within the city to act as seed banks?
I'm not quite sure who the right person to direct that
I
one
think
is.
that
Items, yes.
okay. When we go to the foreign countries, they have some landscape trees like the oranges and also some lemons planted at the cityscapes, and I think that in Terengganu, in Majlis Pembandaran Kemaman, they planted the Garcinia celebica, or previously known as Garcinia hombroniana, the seashore mangosteen, as the landscape trees. And it is also very good to ensure that the cityscape is complementing the GenBank effort in ensure the conservation is there. But I think that the suitability of the species is the most problem, because not all the fruit trees is suitable for the cityscapes. The maintenance and so on. But I think that Garcinia is the most suitable candidate for this kind of cityscape conservation in complementing the GenBank. But I think that, how about from FICUS or from the TRCRC experience? Maybe you can share any other.
Well, I've seen many countries, in fact, not only, well, less in Malaysia, but a lot of countries in even in China, in Hong Kong, and even in Africa, in Cape Town, where they use ficus as part of the, as Dr. Faisal mentioned today, as part of the landscape and also along the road. But what is very important, and I compare this to those planted in Tawau and those planted in Cape Town. Those in Cape Town, they are well maintained, well managed. So it doesn't really cause any form of danger. When you talk about branches falling, you talk about trees falling down. So you need to well maintain them. It is, well, not the best species. If you plant fruit trees, especially ficus, because it attracts also a lot of bats and animals to come into town. And in 2021, we had a program in Lahad Datu during World Environment Day to remove all fig plants from town back to the forest. And it is a good sort of event that we held, because figs in town, they cause a lot of damage to buildings. They block the gutter, they damage whatever you put on the wall, they grow in cracks, they are so resilient. And we had that, and we removed a few thousand figs, wild figs from town, and then we nurse them back to health, and then we move them back to where they should belong, which is in the forest. But I agree, if we can integrate those endangered species with the landscape, it would definitely add to your GenBank. And just to add, in our GenBank, you need to have, I think Paul did also mention, a species that originate from various parts of Sabah. So we have those in our GenBank, some from Lahad Datu, some from Ranau, some from Kota Kinabalu, and all are tagged. So we know which is coming from where. And the other things that I want to also highlight is that some of the figs, the same species, but the fruits are different in size. So one is bigger than the other. So even this is an important sort of, even a small fruit might have other characteristics that's very important for the germplasm. So we also bring in those two and cultivate those. Thank you.
Thank you very much indeed. Do you by any chance know of a similar organization encouraging the growth of figs in Peninsular Malaysia? No.
Not that I know, not that I know of.
Because I think that your message is something which, although we don't have orangutans in the peninsula, we should take to heart here in the peninsula and try and see if we can get something going. Good. Thank you very much indeed.
Dr.
Somebody?
Dzaeman and Dr. Paul Smith would like to contribute to the original question.
Okay. Dr. Dzaeman?
Yeah, I just have one point to make. So in Selangor, right, land is a very limiting factor. We have a lot of very big ambitions to connect our forest parks together. And now we're working on a program with the Malaysian Nature Society called RUGS, which is Rangkaian, it's a connectivity program basically. And in that program, we have mapped out, the key urban parks, Kiara, Bukit Kiara, Bukit Dinding, Bukit Gasing, and we're looking at how can we then start to restore these areas because they were all ex kind of rubber estates and things like this by using high conservation material. So back in 2009, we did a program where we were planting a lot of endangered species in an arboretum sort of manner in Bukit Kiara, and that was quite successful. If you go Bukit Kiara right now, you see them. Well, most of the trees along the road in Bukit Kiara are high conservation value. If you know how to identify them, and also we also then working now with different townships which have environmental pledges as well. So the ESG is now a big buzzword. A lot of them are trying to see how they can work within their own kind of townships to actually promote native species. So a lot of different townships that we have working with are looking at promoting only native, but there are also some looking at also with native and also with conservation value as well. So our office is actually based in one of them in Albina, and over there we're collecting from the forest reserves with all known locations and targets for Selangor, and then returning them back into the urban parks that they have. And with diplocarps is very difficult. They're self-pruning, their branches fall. It's not ideal for roadside trees, and the roots are they get big. They're very big. So you really need to be very careful on where you're planting them, and so that they can actually last and fruit and flower. But if you go to Thailand, for example, there's certain streets in Thailand which actually have full of diplocarps, and in certain seasons you actually see the diplocarps iconic seeds all over the street scapes, and so that's something that we wish to recreate in this area as well. So yeah, thanks.
Good. Thank you very much indeed. Dr. Paul Smith.
Yeah, just a couple of points. Urban trees, native tree species, a good example would be Shanghai in China, where Shanghai Qianshan Botanical Garden works closely with the municipality, and they grow over 100 native species in urban landscapes. One of the issues that you need to be aware of is not only the things that have already been mentioned around branch drop and various sort of other dangers, bringing in unwanted bats and what have you, but also climate tolerance. So one of the problems with cityscapes is the heat island effect. If you've got a lot of concrete, then your city center can be 5-6 degrees Celsius warmer than green areas where these trees occur. So one of the things that we have developed with help from botanic gardens all over the world, and in fact the tool was initially developed by Australian gardens, is the climate assessment tool, which again you can find on BGCI's website. You can choose your location, you can choose your tree species, and you've got 60,000 to choose from. And you can choose your climate scenarios. So is it business as usual, in which case we're getting warmer and warmer? Are we going to get on top of carbon emissions? You can choose your scenario, and it will give you an output as to whether your tree is likely to still be there in 50 years' time or in 90 years' time. Now, the great thing about that data set is it doesn't just use natural distributions. It uses botanic garden distributions as well, and it uses street tree data. So we have street tree data on about 4,000 different tree species. And this is used, the CAT, as it's called, the climate assessment tool, is used as a planning tool around the world by different municipalities and others to make sure that what they plant now is going to be able to survive under not only current climate conditions, but future climate conditions as well. That tool is being improved all of the time. So it's certainly something to keep an eye on. But as we add more data, then the better that tool will get.
Thank you very much indeed. And that brings us precisely to 15:25 Malaysia time, which I think is the time that we're allowed for our discussion. So may I pass you over to the Master of Ceremonies for arranging for the closing summary, please.
Thank you to all the panelists for sharing your incredible insights. To wrap up the day, I'd like to now invite Dr. Gary Theseira, Chair of the Council of CGM, to deliver the closing remarks. Dr. Gary, over to you.
Thank you very much, Avicenna. It's been an extremely informative afternoon. I think I'm not sure that anyone could disagree with the amount of information that we've received and the depth of the discussion here this afternoon. We do know that as a civilization, we rely on plants for food, for fiber, including wood and cotton threads. We rely on them for fats and oils, fuel, feed for animals. And it's not just these agricultural and horticultural crops that we're always concerned about. We're also concerned with the broader biodiversity that we are also attempting to conserve. All the cultivated species that we have and the wild relatives support a huge plethora of organisms, whether they be pests and predators, whether they be pollinators. They also, are banks for diseases, all part of the ecosystem. The wild relatives support insects, arthropods, fungi. As already mentioned, bats. So mammals, birds, and reptiles, even amphibians that breed in the water in whorls of leaves and things like that. So in terms of the value to business, to corporate community, to human civilization, these are really very, very important resources. And understanding how to conserve them and ensure that if they are threatened in any way, that we can bring them back and keep them as a part of our gene bank is very, very important. So we've been very, very privileged to have with us Åsmund Asdal, who walked us through the Svalbard seed bank up there, letting us understand how seeds accessions can be stored there as well. We've had Dr. Dzaeman of TRCRC talking about the conservation, particularly in parts of Borneo, Dr. Norfaizal of MARDI, talking about some of the more important food and fruit species for Malaysia. We've been very privileged also to have who had to wake up very early in the morning, Dr. Paul Smith of BGCI, explain to us the intricacies of keeping alive, in particular, the species that are recalcitrant. And finally, Dr. Zainal of BORA, who shared with us a great deal regarding conservation in Sabah. I think all of these have been extremely rich, and we barely scratched the surface this afternoon, but I think all in all, it's been an extremely rich discussion. The idea, obviously, is that as an administration, as a government, as a country with mega biodiversity, we really need to understand our biodiversity, and we need to think about ways in which we can ensure the continued sustainable use of this biodiversity, coexisting with it, benefiting from it, and benefiting from its richness, and in fact sharing this richness with other parts of the world that are able to accept it. I'd like to also thank our own Dr. Serina Hijjas, who provided the opening remarks for this session, and Dr. Barlow, who has very ably moderated this session. Of course, in the background, the members of the Secretariat, thank you so very much for your help, and Avicenna for emceeing this program. So with that, we call an end to this webinar, Seeds of Tomorrow, Addressing Climate Challenges with the Svalbard Global Seed Vault. We hope that you will join us here at Climate Governance Malaysia for our future webinars on topics like this, where we do consider the impact of climate and the role of governance in not only surviving and being part of addressing climate resilience, but also finding ways to prosper with respect to the changing climate, even as we work on reducing the climate impacts and the release of greenhouse gases. Thank you very much for joining us. Do join us next time. Thank you. Have a pleasant evening.
Thank you, Dr. Gary, for delivering such a great conclusion to today's event. Ladies and gentlemen, with this, we officially conclude the Seeds of Tomorrow, Addressing Climate Challenges, the Svalbard Global Seed Vault. We'd like to thank all our participants for joining us today. Before you leave, please take a moment to complete our feedback survey, as your feedback will be most helpful in shaping future initiatives. This session has been recorded and will be made available on the Climate Governance Malaysia YouTube channel, complete with time-tagged content and accompanying blog posts. We invite you to stay engaged with us through our CGM newsletter and our official LinkedIn page for continued updates. On behalf of the organizing team, we extend our heartfelt gratitude to our distinguished speakers for sharing their expertise and to all participants for being part of these vital conversations. Together, let us continue driving change and shaping a more sustainable future. Thank you and see you next time. Have a good afternoon.